In this video, we're going to talk about the organic compound group proteins. Now that we've already learned carbohydrates and lipids, you shouldn't be surprised to see that proteins contain hydrogen, oxygen, and carbon. But in addition to that, they will also contain the element nitrogen. This is a good giveaway for you when you're looking at a compound, because if you see nitrogen, you're probably looking at a protein. The monomer of a protein is called an amino acid. The name amino acid comes from the groups, the functional groups that are found within this compound. We'll talk more about functional groups on the upcoming slides, but for right now, the two functional groups that you should be able to identify in an amino acid are the amino group and the carboxyl group. Sometimes carboxyl groups are called carboxylic acids, so that's where the name amino acid comes from. The amino group will always be a nitrogen with two hydrogens attached, and the carboxyl group will always be a carbon double bonded to an oxygen and then also bonded to an OH. So the amino acid should be very easy for you to build and identify in your lab work. In addition to always having an amino group and a carboxyl group, an amino acid will always have a hydrogen and what is called an R group bonded to that middle carbon. And the R group is just a variable group where we can sub in any of 20 different variations to make different amino acids with different properties. So that's the basic structure. If you link two of the amino acids together, the dimer will be called a dipeptide. So that, again, is just the same condensation reaction where one water is formed. And if you link multiple amino acids together, you get a polypeptide. There are two types of polypeptides. You want to make sure you write this information down on your graphic organizer. There are both fibrous and globular proteins. So initially, when you get a long chain of amino acids, just all the amino acids linking together, they start off as this long chain, and that would have the structure of a fibrous protein. An example of a fibrous protein would be something like your hair or a spider web, a long, thin chain of amino acids. But there is the possibility that the protein chain, the polypeptide, will fold upon itself and become more of a blob, and that's what we call globular protein. The most common example of a globular protein is an enzyme. We've talked about those a lot this year. Another common example of a globular protein is hemoglobin, which is seen right here. And you can see how that amino acid chain has been folded all over on itself to create the perfect molecule for carrying oxygen through your body. The two main functions of proteins are to build your body tissues and to work as enzymes in your body. But proteins probably are the most diverse in function of any of the organic compound groups and really can be used to do many, many things in your body. So here's a little bit more about functional groups. We've already said that amino acids are very easy to identify because they contain always an amino group and a carboxyl group. In organic chemistry, there are many different functional groups. Some examples here are hydroxyl group, phosphate groups, R groups. They're just groups of atoms that we see over and over again in different compounds, and the different groups can have different properties. We're not asking you to memorize the properties of those functional groups. We just want you to be aware of. So here's some of the names of the most common functional groups, the ones that you'll see and build throughout our labs. And just so you know the structure, it's listed there for you. You are not expected to memorize this. Again, really you just need to zone in on the amino group and the carboxyl group that are found in the amines. Here's, instead of in words, the exact same information, but just in pictures, so you can see what these functional groups look like. When you built your lipids, you had hydroxyl groups. When you build different molecules, you'll see these carbonyl groups, carboxyl groups, amino groups. The phosphate group is present in ATP, something called a methyl group right here. Again, don't memorize, but you do need to be able to recognize an amino group and a carboxyl group when you see them in these amino acids. In the lab, when you want to test for protein, this is another simple test. It doesn't take a large volume of the solution, and you do not need to heat it, so you can just use the spot plate. The test is called Biuret. You're just going to add equal parts of your test solution with the Biuret indicator. Biuret starts off looking like this, a light blue color, and if protein is present in the test solution, it will turn a lavender color. Once again, I really encourage you to run a control test. Sometimes in the lab, it's difficult to tell the difference between the light blue and the lavender, so it helps if you always run a negative test with just using water, so that you really can see the difference between the light blue and the light purple color. So now that we've come through and we've done lipids and we've done carbohydrates and now we're doing proteins, you should be starting to get comfortable after having built them in the lab, identifying which types of organic compounds these are. So if you want to, you can pause the video and you can try to take a guess of what different organic compounds you see here. But I'll walk you through how I would identify these. The first thing that I notice in this molecule here on the left is that there's definitely a ring, a hexagonal ring-shaped structure. And for me, that's the giveaway that I'm looking at a carbohydrate. And I also see a big similarity between this molecule and the molecule over here on the top. I see again, ring linked to a ring, linked to a ring, linked to a ring. This looks like a lot what we did in the carbohydrate lab, where you built a bunch of glucose molecules and then had condensation reactions occurring between them where water molecules were lost in each bonding. So this here is a monosaccharide, where this is a polysaccharide, but definitely they're both carbohydrates. This molecule, if you've been paying attention to the lecture, should be very easy to identify because we see those nitrogens. One thing to notice, even though it's a little cut off in the picture here, it's the exact same type of bond that we did in the last lab. So you're still doing a condensation reaction with water coming out, but when those amino acids bond together, we will call that a peptide bond. I'll write it over here since it's a little cut off. Peptide bond. It's no different than any other condensation reaction, but because we're bonding peptides, making a dipeptide or a polypeptide, we give that bond a special name. Last but not least, down here at the bottom, this should be very easily recognized because you guys built this in the lab as well. When I look at this molecule, I basically see the letter E. I see a glycerol backbone with 3 fatty acid tails, so this is a triglyceride, which would fall into the lipid organic compound category. One final thing that we have to talk about in our protein video is enzymes. One of the most important things that proteins do for us is acting as enzymes. So enzymes are globular proteins that can speed up reactions or lower activation energies. This is really important in biology because sometimes we need reactions to happen either quickly in our body or at a lower temperature. You can't light a Bunsen burner inside your cells to make a reaction occur. So you need these enzymes in order for many of the reactions in your body to happen. They're very easily identified because when we name enzymes, we always end them with ASE, so maltase, amylase, carbonic anhydrase. The name of the enzyme usually is related to what it works on or what it breaks apart. So maltase would work with maltose. Amylase would work with amylose. Enzyme function can be affected by temperature and pH, so we want to talk about that briefly. If I look at the activity of an enzyme, and I graph that over pH, or if I look at the activity of an enzyme, and I graph that over temperature, the activity is going to be very specific. So, for example, enzymes in your stomach will function at a pH, I guess I didn't label this here if this is 7, that is acidic because our stomachs are acidic. Whereas enzymes that are in your small intestines would work at a pH that was more basic. The same thing for temperature. Enzymes are made to work in specific temperatures. So if we were graphing, and let's say we have 100 degrees over here, 0 degrees over here. Most of the enzymes in our body would work well over here in this warmer temperature because our bodies are 98 degrees. But if we were to look at the enzymes, let's say in a tree, their enzymes might have a peak activity rate at a cooler temperature, especially if they're from the Midwest and they're used to surviving through the winters. Enzymes can be reused over and over again by your body. Remember that these are globular proteins that can speed up chemical reactions or just lower the activation energy to get a reaction to happen. Enzymes are super specific. They really work like a lock and a key. The substrate, or whatever we're trying to work on will fit directly into the enzyme in what is called the active site. So this vocab term right here, active site, would be this spot right here in the enzyme. And you can see, just like pieces of a puzzle, how the enzyme and the substrate will fit together. In this case, it looks like we're showing a hydrolysis reaction where some kind of dimer, maybe it's a disaccharide or a dipeptide, is being broken down into monomer units. This reaction maybe would happen over a long period of time or maybe at a very high temperature, but by using the enzyme, it would happen quickly or at a lower temperature. Take a second to think about the concepts that we've talked about in this video and make sure you can answer these questions. Again, you might want to pause the video to think about this. But I'll go ahead and walk you through the answers. What do enzymes have to do with activation energy? So remember, enzymes lower activation energy, so you don't have to have a Bunsen burner for every single biological reaction. And are vitamins an organic compound? This one, the answer is actually no. Vitamins are not organic compounds, but if you ever stop to wonder why you take vitamins, vitamins are called coenzymes. They're not actually enzymes, but they work with the enzymes. I'd like to think about vitamins as like a WD-40 for an enzyme. Just like you'd spray WD-40 on a squeaky door hinge to help it move better, enzymes sometimes need a little help to work more efficiently. So the vitamins can work in that active site to help the substrates click in and out faster and more efficiently.